Keep
so
it
much.
short.
Thank you for organizing this wonderful, really creative webinar with speakers from all over the world on, on otology. So welcome everyone. This is Dr. Judith Kempfel. She is a clinician scientist at Harvard Medical School and a researcher who focuses on regenerative approaches to improve inner ear hearing loss and using techniques of cellular reprogramming, gene therapy, and small molecule drug development. She has a special interest in Schwann cells and glia of the inner ear and auditory system and their role in repairing hearing restoration. Dr. Kempel also treats otologic disorders in pediatric and adult patients, including hearing loss, chronic ear disease, skull-base disorders, balance disorders, and vestibular schwannomas. Thank you so much, Dr. Kempel, for organizing this, again, really creative, interesting, and collaborative webinar. And thank you to all of our panelists who are joining us from all different time zones.
Well, thank you, everyone. It's my great pleasure to serve as your moderator tonight. This is a webinar that's hosted by our ARO Clinician Scientists Committee. It's titled The Global Otologist, Funding, Partnerships, and Innovation Across Borders. And we're very honored to have an exceptional panel of clinician scientists we've heard from all over the world, where hopefully each of them has a unique perspective on their experience and how to initiate or to facilitate successful collaborations, both in academia and then in industry as well. Our panelists tonight are Stephen O'Leary, a professor of otolaryngology at the Royal Victorian Eye and Ear Hospital at the University of Melbourne in Australia. It's morning for him there. Good morning, Stephen. Then we
Morning.
have Yen-Fu Cheng, professor of otolaryngology at the Taipei Veterans General Hospital and the National Yang-Ming Chiao Tung University in Taiwan. And special thanks to Professor Hubert Löwenheim from Germany. He's a professor of otolaryngology at the University of Tübingen Medical Center and Eberhard Karls University in Tübingen. For him, it's 1 a.m. at night. I'm very grateful that he agreed to participate. And then last but not least, Professor Heinrich Stecker, a professor of otolaryngology at Kansas University Medical Center at the University of Kansas here in the US. And this evening's discussion should focus on the landscape of international collaboration in otologic research. And as we all know right now, we have challenging times as it comes to research funding, especially in the United States. And this makes it all the more important for us to look outward to international partnerships or industry collaborations as a way to share our expertise, to pool the resources, and hopefully to push the field forward. And tonight you'll hear about funding opportunities, examples of translational projects, some that have worked or may not have worked, and some practical advice, hopefully, especially for our junior clinician scientists, on how to build such a global network and how that can make a real difference in your career in the future, hopefully. And to get us started, I'm going to ask each of our panelists to take about 2 minutes to introduce themselves, highlighting their clinical and their research experience, but also their connections with industry. And after that, we'll move right into the discussion. Thank you all for being here. And with that, let's kick things off with Professor Stephen O'Leary. Stephen, please.
Well, hi, I'm Stephen O'Leary, and I started my life as a disgruntled junior doctor, I think it's probably fair to say. So I decided to do a PhD with Graham Clark, who I'd done an undergraduate degree with as a medical student, and ended up landing at the year that commercial cochlear implants really came of age. When Cochlear did its first commercial implant, I was there, and I was given an electrophysiology project, basically current spread within the cochlea and how do you work it out in animals. From there, I maintained an interest in electrophysiology, but after becoming a surgeon, became particularly interested in operating on the inner ear without causing damage to it, with a view to improving cochlear implant outcomes and also ultimately, of course, maybe for regenerative therapies. And I guess that's where I've spent most of my career, in that space, building actually on the electrophysiology that I started off with and utilizing that to try to improve implant outcomes. So the other major thing I did early was to look at drugs to protect the inner ear. And I was lucky enough to choose dexamethasone in our experimental work, which has, of course, gone most of the way through the regulatory process for at least, a couple of companies now, and hopefully will be in our patients very soon. So I've been part of that journey since the first experiments in the laboratory. Another thing I've done that hasn't worked so well, but has led me into artificial intelligence much more, is surgical simulation. I mean, I've done a lot of work in that space and surgical simulation and training, but that hasn't been successfully translated as opposed to some of the other things I've talked about. However, it's a good story of how that kind of enterprise can lead off into other areas that then become precedent in their own way, like artificial intelligence.
Thank you very much, Professor Leary. That was great. And let's move on to Professor Yanfu Cheng.
Hi, Judith. Thank you for the invitation, and hi everyone. I'm so honored to be able to be the panelist among all our masters that I met regularly recently. Yeah. So I got my research training in Boston, and actually together with Judith from day one of my research career. We share the same bench actually. Yeah, and then after my residency, by the way, I'm an ENT by training. And then after, I'm currently a physician scientist at the Veteran General Hospital and also professor of otolaryngology and also at the Institute of Brain Science. And I'm currently leading a research center of gene therapy in my university and the hospital too. And so during my research training, as I mentioned earlier, that I was actually sharing the bench with Judith and under the supervision of Dr. Albert H. So my research was about approaching the stem cell strategy for inner ear regeneration. And then during my postdoc, back to then the CRISPR has emerged. So I decided to explore more on the genetic approach for hearing regeneration. And then when I returned back to Taiwan, I started my career. I decided to have a genetic path for the hearing regeneration as my research topic. So my current research focus is on the genetic deafness, and then which we do clinical and basic research on genetic diagnosis, natural history, and also gene therapy for the deafness patients. So my experience with the industry is that I do have some collaboration with some of the biotech that has been recently merged by the big pharma or the bigger biotech recently. So I do some ongoing research with them currently. Thank you.
Thank you, Yanfu. That's wonderful insights. And next up, we'll hear from Professor Hubert Löwenheim.
So, hello everyone. Thank you all also for the kind invitation to this very interesting webinar. I started out as a first-year medical student attending a physiology lecture, and the lecturer was a professor called Rainer Klinke, who was a famous auditory physiologist at the time. You could call him the Nelson Chiang of Germany. And yeah, he gave a great lecture, and I got hooked up to auditory physiology in an instant. And since then, I've stayed with that. I picked up a doctoral degree in the laboratory of neurophysiology with Professor Klinke. And after I finished my medical studies in Frankfurt, I transferred to Tübingen to a research heavy department of otolaryngology, had a neck surgery with Professor Zenner. And I wanted to continue in auditory physiology, but he kind of transformed me into a cell biologist. And he told me my task would be to regenerate the hair cell. That was quite, I would say, some decades ago, and I'm still trying to regenerate the hair cell. So, heavy tasks can take a long time. Along the way, after completing residency and going through the career path, I founded my first company actually at the end of my residency. The company was called Otogene and has transformed into Sound Pharmaceuticals, which some of you may know, which still exists. And 12 years ago, I founded my second company called Acusia Therapeutics, which is involved in developing a potassium channel agonist for improvement of hearing. So also quite some translational and industry connection in my career.
Thank you very much. And finally, Professor Heinrich Stecker.
Judith, thank you for organizing this. I think the path for surgeon scientists is so hard. So I think this is an important, really important evening to get everyone together. So I started out completely differently. I went to medical school because I was interested in infectious diseases, and ended up interested in virology. And because I was interested in music, got detoured during medical school to ENT. And so my interests fused, and really became interested in gene therapy and hearing disorders. And that's sort of the career path I followed for 30 years or so. My PhD thesis was actually in a spinal cord injury lab in Belgium, studying neurotrophic factors. And I was initially interested in improving cochlear implant outcomes using gene therapy to deliver neurotrophins. So that's sort of how I got started. In my academic career, really, the corporate side has always been important because it is a way for more directly getting therapeutics to patients. And I think we can argue this back and forth, but I think as a clinician scientist, our role is to stand between the basic scientists and the pure clinicians and really bridge those two areas. And I've worked with small gene therapy startups that have now morphed into Precigen, and got to really take the first inner ear gene therapy program all the way from the discovery side through human clinical trials, collaborating with Novartis. And then I've started my own gene therapy company, which hopefully we will have a product that will be tested by Hubert at some point, because he has the patients. And it sort of links we've built up over the years, I think we'll discuss this as we go on, are really, really important for developing our programs.
Thank you all for the introductions. I think I'd like to move to the discussion part right away. We'll go through a few key questions that I have, and then for the audience, feel free to add your own related questions in the chat as we go. I'll try to field them all, and depending on the time, we may have the opportunity to have a more open discussion toward the end. And to start, I'd like to talk about funding. My question to the panel is, are there any unique non-federal funding mechanisms you've used or that you know about that specifically support international or industry academic collaborations? And are there any that are specific for early career investigators? Heinrich, maybe I'll ask you to begin here.
So when I was first an attending at the University of Maryland, I was approached by a company, Genvec, that wanted access to inner ear models. They had licensed the intellectual property for the atonal gene. And we actually had some corporate money and got a lot of our startup from a state, so a United States state-based funding mechanism to support local biotech. So it was called the Maryland Industrial Partnership. And it was actually a very decent amount of money, equivalent to NIH funding, that funded 3 years of startup money that took us, produced a lot of preliminary data that allowed us to really access the SBIR programs for the NIH afterwards.
Okay, great, wonderful. Hubert, how about you? How about in Germany?
Yeah, I mean, on the academic side, we have these European Union grants that actually do allow to include American laboratories. So we did have one or two programs that not only included European sites, but also American sites in Boston and Stanford, for example. I hope that, you know, the cooperation between NIH and EU will continue on that front, so that this mutual funding will still be possible. On the translational side, when I founded my first company, we had identified a drug target, and it was clear that this could not be funded through academic funds on its own. And we had already established an EU-US academic collaboration between Tübingen and at that time Seattle. And that led to a transnational funding or a foundation of a company that was funded by venture money, in the beginning seed money, and that was matched by so-called matching funds from the state where I live, that's Baden-Württemberg, and also the federal state of Germany. So and these funds actually allowed also to subsidize the American subsidiary from the mother company that was based in Germany. So there was quite some flow of money. In total, the funding over a 4-year period was 40 million, so quite substantial amount. But you know, that's kind of necessary if you go through a drug development program. This is possible. You need the right partners, you need the right network, and you know, if you really want that, you can do that even at young age. As I said, I mean, I was just at the end of my residency, so around 30 at the time, and actually I took some time off. After the September 11 events in the year 2001, we had to transform the company. As you know, it's now Sound Pharmaceuticals. And I decided to go back completely to academia after 4 years of being in the biotech scene, and founded another company then 10 years later. And quite frankly, having been going through that process one time really helped me to do it a second time. I don't think I would have done it a second time if I wouldn't have had the experience of the first time. And we have repeated a full-fledged drug development program that is now in a phase 2 clinical trial, and also needed a lot of funding in the range of 25 million. I have a more passive role in that company because I have a clinical job to do, but you can do it as a founder. So we had an academic idea, we transformed it, and were able to do the translation.
Great. Yan-Fu, what was your experience in Taiwan?
Yeah, so I think so in Taiwan, in general, the grant is pretty small. So I heavily rely on the intramural funding from the hospital and the university in the very, very beginning. And before I get my first R1 equivalent federal grant in Taiwan. But then afterwards, after one or maybe after 1 year, actually, when I just started the lab, and then there are some industrial opportunity. Then so I have a collaboration with the two major hearing-related biotech, and then that really gave me a big boost to my lab. And also I try to, because as a junior investigator, I try to join some of the big umbrella grants in Taiwan, because my interest is about genetic hearing loss, and then some of the genetic disorder related grant that I have a good connection with. So I joined them and then explore the part of the hearing. And also because I'm having some so-called international collaboration grant from our National Research and Technology Council. So I am having I was just in Lithuania last week to have some collaboration meeting with my partner before after I went to Tübingen for the inner ear biology. And also I'm having we are writing a collaboration grant with Heinrich to partner with some of our German partners. So I think those international and industrial collaboration are also very are actually important in my research career so far. Thank you.
Great. Stephen, how about Australia? How about you?
Yeah, like most of, apart from our government grants, we're lucky to have access to a fund called the Garnet Pass and Rodney Williams Memorial Foundation, now the Pass Williams Foundation, and its mantra is to actually promote excellence in otolaryngology. And they gave me a fellowship to go overseas to Europe, where I worked with Jan Veltman. And when I came back, I've had fairly continuous funding from them. These grants are what you might call enough to keep about. These used to be a whole person employed, but now it'll be about half a person, unfortunately, the way things are going. And it meant that I could start to develop ideas myself and bring them into the laboratory. In the broader context, of course, Graham Clark was still working, and I was in the fold of his laboratories. And we had at the time a partnership grant between the government to create like a small commercial entity that worked very closely with companies, one of which was Cochlear. Now, I didn't actually benefit much directly from that, but it helped develop the relationship with my university and Cochlear and keep that alive, and through significant funding. So by the time that some of the ideas that I'd been working with in the laboratories, particularly in terms of drug delivery to the inner ear, and later in terms of intraoperative monitoring, became interesting, there was a fairly open door to talk to Cochlear about them and to have, really quite a close dialogue. And it was later that more direct commercial funding came through Cochlear to help boost those programs on. The RNID is the other one that I've certainly really been very grateful for the support from. You probably know they're in the UK, and they do allow international collaborations. And I know that at least several people on this panel have had their funding as well. But all of this helps, particularly when you're younger and you're trying to define your research space and find something unique and different that you can offer. And I think that's really important as in your earlier research career. I mean, my approach was to try to have several things going at once. I was lucky enough to find the funding for that and to find people to work with for that. Not everything worked, you might say, in terms of coming through to commercialization, but having multiple areas that might work was certainly a good strategy, I think, so that you would just drop some and you would pursue others according to how the clinical translation and opportunities seem to be going. I've never had success with international grants that link up some of the major funding organisations. I mean, we do have one between Australia and the EU funding, but the difficulty is that you have to be on the initial grant in the EU as a named investigator, and then you can actually access considerable money in Australia. But of course, it doesn't quite work that way. I mean, unless you actually have collaborators and they think you can help from Australia without sending money to Australia, then you're not likely to get onto the grants. And by the time you've found synergy with your colleagues, because they've got significant funding, the opportunities pass. So that's a bit of a catch-22 for that funding stream, which I find a little frustrating. But it's worth looking at your national funders for specific grants of that nature, because sometimes they exist, and sometimes if you have the right conversations in advance with international colleagues, you might be able to offer something, get your name onto the grant, even if there's no funding coming to you, and then there might be an opportunity locally to then access bilateral funding through leverage, basically.
Thank you. Yeah, that's great. When you think back at your very first international collaboration, what advice would you give a junior investigator who wants to initiate one, or how did you get yours started? Hubert, maybe you want to kick that one off.
Yeah, regarding the second question, right? Yeah, I think to initiate your clinician scientist career, it's really very important to find a mentor who really gets you started. In my case, we designed an idea about what would interest me and what would interest the department, and from there on, we made connections to leading international laboratories. So, for example, in order to get my methodology off the ground, I visited labs in the US and the UK. So, for example, I went to Jeff Corwin's lab in Charlottesville, and also to an electron microscopy facility in the UK. So I had the chance to really improve or enlarge my methodological capabilities, and then from that on transfer those into a very small laboratory. I still remember that. It was like 8 square feet or square meters in size, and then just grow from there, develop my own ideas, and then find scientists. And I mean that from I think that's also very important, that when you are a clinician scientist, you also need to find mentors that are pure natural scientists that do that full time. And they can actually give you insights where so you're not wasting your time on maybe false pathways. And I think you should try to get their advice, try to have both clinician scientist mentors, but also full scientist mentors to help you find to follow your ideas. I think that's key to get off the ground.
Yeah, very important. Heinrich, how about you?
Well, I mean, it helps if you have personal links to begin with. So I'm German. I did all my medical training in the US, but I did my PhD in Belgium. And based on that, when my lab friend Philippe Lefebvre, who's the chair of the ENT program in Liège, was hosting inner ear biology, I went to inner ear biology for the first time after really only attending AROs for years and years, and was approached by a German otolaryngologist, Mark Pretorius from Heidelberg, who was interested in learning more about gene therapy, who then came and visited my lab in Baltimore. And that sort of set up a collaboration between my lab and University of Heidelberg, where we had fellows from Germany come over regularly. And that's sort of what really got my international collaboration going. I'd credit Mark with that. So I think it's very important to look outside of your home base, see what's going on in other countries, and attend meetings widely, and then you'll meet kindred spirits, right? So there's not that many of us worldwide. So I think you do need to cast your net widely to meet people that are interested in the same thing, or the same, philosophically the same thing.
Right. Yes. Yeah, Yen-Fu, how about you? I mean, obviously,
Yeah.
know your time here.
Yeah. So I think the advices that I want to give is the connection and the networking. Yeah. So it's like Heinrich. So I did my ENT residency in Taiwan, and then I decided to expand my kind of my own network or the knowledge. So I went to Boston for my PhD and the postdoc training, and then over there I learned so many smart and hardworking scientists. One of them is Judy. So, and then I learned how to do the research because before I had a zero experience in research, and I started to after I finished my PhD, then I met a very famous viral vector expert, Dr. Luke Vandenberg at the Department of Ophthalmology in the same Mass Eye and Ear Infirmary. And then since then, I established a great friendship with him, and we met like regularly, and he became my mentor since then. And then I realized that I mean, the hearing will be the genetic period since then. So, and he helped me a lot. And then after I come back to Taiwan, and back to then, I'm making vectors and establishing some animal model are really expensive and require some domain knowledge. And he was kind enough to offer me some of the very cutting edge vectors. So, and then we published a couple papers together. Yeah, so I think he's I mean, brought to approach some people or researcher in your field and then meet them regularly are the keys. And also I think to attending like a conference like ARO or the Academy conference also helped a lot, because you could meet many people. As Heinrich said, that you might be interested in a small field of research, and then over there you find people with the same interest with you, and then you can talk, and then you can establish some collaboration projects over there. I think it's very important. So I would say connection and networking. So try to expand your network. All right.
That sounds good. Stephen, how did you get started?
Look, I was really lucky to have a mentor. That was Graham Clark, and that also was a very multidisciplinary environment. And so from my earliest research experiences, I've seen what could happen when engineers work with audiologists, work with clinicians, right? And of course, designing cochlear implants, understanding them from how they work was one of the first things I was interested in, way before the surgical aspects. But, you know, I guess I was always immersed and surrounded by people from these disciplines, much more broadly than ENT. And look, I guess I had an attitude at the time, and it was easy perhaps in that field because it was only just emerging, and the basic questions were not answered, like how do we actually code a cochlear implant, for example, that I really made a very active effort to talk deeply with engineers and audiologists, and actually thought it was my responsibility and job to understand everything about this field from the ground up. I'm sure that's given me a really deep sense of understanding and a sense of a language for dialogue with people beyond the clinical space. And I think that's been really helpful. And then, of course, I echo all the sentiments about going to meetings. In addition to ARO, which has always been one of my favorite meetings, particularly just going around the posters, of course, it was, you know, the Asilomar meetings, which was the cochlear implant meeting where you get all the engineers and all the audiologists, and you'd also get a few clinicians, right? Who would listen, and you would be bamboozled by a lot of the engineering, but you know, my advice is just hang in there and learn as much as you can from the scientists about the science as possible. So I think probably my first collaborative papers were on single unit electrophysiology data, coming and discussing with some of the doyens in the field, like Klinker, like Eric Javel, like Nelson Kiang, and then working on data together, right? It was only much later in my career that really rich international collaborations came, but that was by the time that I had something unique to offer, you might say, to the field and could see a translational path, right? And at that point, it's much easier to talk to your clinical colleagues and to start to build collaborative interests in terms of, you know, trying different things out to improve the clinical benefit of the patients. So you could say that my international clinical collaborations came after years of more basic scientific endeavor and research, really with an ear open to a broad number of people from many disciplines, all really interested in improving hearing outcomes, largely through cochlear implants.
Thank you. Well, to all of us on the outside, you all seem extremely successful and accomplished, but as it always goes, not usually everything goes smoothly or runs smoothly. If you look back, was there one common pitfall or a challenge that you faced in one of these collaborations, and how did you overcome them? Could you give us an example? How about Yin-Fu? You want to start?
All right. Yeah, I think the challenge would be, you know, a physical meeting, right? Because everybody knows that the key to success for collaboration is like a communication and a regular meeting, and then share the progress of the project, and so yeah, so and also, you know, if you can apply for a common grant, and then that will tighten the connection better, because you get the pressure to finish some of the milestone. That would also help. So I would say regular meeting and the discussion are the key. And then, of course, we do have this one, we have a very good online meeting system. But, you know, attending like ARO or the inner-ear biology would really help you to have a like more tight connections, and also more opportunities to talk. And then I think that is the most important part, and to have a good connection with your partners, and then communication and discussion regularly. That would be my suggestion.
Thank you. Heinrich, any challenges?
I think it's Well, collaborations are can be challenging anywhere, but especially internationally, there are different requirements different countries have for academic advancements, and things such as impact factor in publications that affect your collaborators, the amount of money the university gives to your collaborator. For example, in Europe, some of the European programs count impact factor publications towards the funding a department gets. And I think it's important to get all of those type of discussions out front so you can set expectations properly. Sort of like dating, you want to set expectations properly ahead of time. And when when, especially if somebody sends you somebody to come work in your lab, there can be expectations of certain publications and stuff, and you need to be aware of that. And if you can honor that so that you can have an honest relationship, I think is the easiest way to explain it.
Yeah, I think that's very important. Stephen, how
Yeah,
about
look,
you? Paul?
Look, I would like to firstly echo Henrik's comments. I mean, I've certainly been in this situation where, I've been really done the majority of work on projects where from the very outset, the authorship's been set, right? And it's been agreed to. And at the end of the day, everyone is happy, okay? I think some of the biggest problems I've seen is when, not necessarily my personal experience, I'm happy to say, but certainly some of the people that I've had to mentor, where they have insisted upon a different position in an author, in an author ordering than another person who's perhaps done more of the work. It typically happens with students, right? And I've seen relationships completely destroyed. And in fact, people standing in departments destroyed that way. So I can't really emphasize enough the importance, as Henrik has said, a bit like dating, getting all of the facts, understanding the context of the individuals, right, in another country. I've written papers and I've been maybe agreed to be the last author, not the first author, because I know how much it means to the collaborator internationally, right? And that's not a problem. Actually, I don't have any issue with it at all, because it's understood up front. And you can be a bit creative about how you attribute attribution. I mean, you can be a corresponding author, perhaps not a first author. There are various ways around it. You just need to know what the tricks are. The other thing I'd just like to say about collaborations is that, we're here talking about funding, and that, of course, for me, some of the most painful moments have been when the funding finishes for something I'm passionate about. Or when someone else gets the funding for an idea that you've thought of and that you've been having grants in for years, and someone else ends up doing it, right? Now, I've come to realize that if the idea was so good that you had a great idea and a grant, that you're probably not alone in that thought, and that's okay. But if someone else makes something fabulous of it, it actually is still okay, because you were right in thinking that was a good idea. And it's taken me a little while to come around to that mature understanding, but I think it's actually a really important thing. A good idea is more important at the end of the day than anything else. And we seldom have them as individuals. The other thing I'd like to say is that there's certainly, you've got to often keep hammering away at a good idea for a long, long time. I've been trying to get funding with Henrik now for about 4 years on something which I'm still very passionate about. I'm hoping this year might be the year. But look, happily Henrik has agreed to be named on our grant. And, it's a bit like playing a game of soccer, right? You have to keep shooting at the goal. And hopefully what you're trying to shoot for isn't absolutely core critical to your survival as a researcher. But it's good to have these kinds of ideas and to keep hammering away at them, keep shooting for goal, because you're only going to get your grant if you keep shooting, basically.
Yeah, I agree. Hubert, any important lessons? You're still muted, sorry.
Well, I would like to spread some enthusiasm about initiating collaborations. I think especially in the hearing field, there is an extremely friendly atmosphere, and I can't really remember that I was ever turned down by anyone in the field, be it in Germany, Europe, internationally, if I asked for something. Visit a lab, learn a methodology, and now in my position, send somebody to a lab to learn something, send somebody to do a postdoc, to do a PhD, or vice versa, people asking our lab to come for a PhD. So I think in general, the field is very open to these collaborations. And I would like to strengthen that, have the courage to ask. People rarely will put you down. If they do, it may be circumstances that they just don't have the capacity, but they will always try to help and maybe recommend another lab that can help you out. So I think there's a very collaborative atmosphere in the field. So just go ahead. Now, and I can only repeat what Hinrich and Stephen have said about, let's say, when you really invest your time, let's say a postdoc, 2 years, right? Let's say you go out of your residency, or you do it after the residency, and you really take up a postdoc to strengthen your scientific background, like we've heard from all of you. It is very important to really discuss with your mentor that may be in another country what the expectations are, because as Henry stated, the systems are different. There may be different expectations with different cultural backgrounds, and usually there are also expectations from not only the individuals that may come to you as into a lab, but also from the sending institutions. So like the Japanese system has certain expectations when they send out people and they go back to their home country, or in other countries. So that is important to know. And also the funding, for example, in Europe, when we apply for a grant, we have to write reports on what we actually applied for. And that is pretty strict. So we have to really stick to the plan and report the plan and then put out publications according to the plan. Whereas in the US system, that is a little more relaxed. You apply for a grant, then you get your money, and then you have a little more freedom to do with the money. Maybe use it for another project that may not be possible in Europe, for example.
Yeah, that's true. Well, thank you all for sharing your experiences. I want to use the last 10 minutes or so that we have to ask the audience if there are any questions. I don't see anything in the chat, so you can just unmute and ask a question to our panelists here.
I think everyone's eating dinner.
I can ask a question. Well, first, thank you, Judith, for putting together this panel, and all the panelists for giving such sound advice, especially those in Europe. It's probably 2 a.m. in the morning, so thank you for staying awake.
Okay.
So what are some challenges as a clinician scientist, as far as pulling away from the clinic side in your country? Like, is there more and more pressure to be more clinically productive and has less time for research?
Maybe I'll tackle that from the US side. So surgeon scientist is particularly difficult because as you build your practice, it's not like you have a week where you're ward attending and then 2 weeks in the lab. If people send you surgeries, you have to take care of them, at least in our system. So in the end, to protect your time, you have to have a system for taking care of both, and that often means, and when I talk to our MD PhDs who are interested in surgical careers, is often accepting a lower salary for systems where your income is based on how much you operate, right? So if you love the research, you have to be willing to make that trade-off.
I could answer it from the Australian perspective. We're quite lucky, I think, in my system, in that our clinical commitments are on a sessional basis, right? They're on a half-day allocation, and a lot of people in our system will work in several public hospitals rather than just one. I mean, now I've committed to one because I want to be in with my laboratory, but and the hospital actually allocates not to a service, not to an individual. So I've had protected time for research that's paid by the university at a much lower rate than the clinical work, admittedly. But in our system as well, what private practice we have, and I do it 1 day a week, I'm actually doing it today, that actually tops up the income. So protected time, accepting, a reduced salary, but, from the point of view in Australia, it's just, do you want 2 Porsches and 3 holiday houses or not? I mean, it's that kind of decision. It's not, are you going to have a good life? And in fact, when I see a lot of my colleagues retiring, before me because they're jaded and sick of full-time clinical practice, I think, aren't I lucky? Because I'm never bored, I can't underestimate what it means to have something that you think about every day of your life, right? Which I've thought about implants every day of my life, and wrote up this morning and wrote a few hypotheses and things. I think, will I get to test this one? Literally, okay. And that keeps you young and fresh, and I can't underestimate what it means to be working really well with younger people throughout your career. Now, I get this in the hospital, but I also get this very much in the laboratories, and it's a joy, and it keeps you fresh, and you won't get burnout, I trust, with that kind of approach.
Yeah.
Any other comments, other questions?
Yeah, so I may comment from the European side. Yeah, I think every system has its own way to acknowledge that the clinician scientist is in a particular situation. And I see Aaron Riemenschneider in the audience, and he gave a great talk at the ARO, I think last year, about the difference between a clinician scientist and a surgeon scientist. Hinrich actually used that term. And surgery or being in a surgical department makes your life more unpredictable than if you were, let's say, in a conservative department where a clinician scientist would be situated. So that is a very particular situation. And my recommendation is, if you plan your time as a surgeon scientist, do it sequentially rather than in parallel, because as I said, surgery is unpredictable. So if you plan like a half-day surgery and a half-day research, most of the time your research half-day will be killed because there's a complication in surgery or somebody else needs you in surgery. So that's not working. It's much better to say, okay, I'm a month off, no surgeries, and I do my research project in that 1 month. So that is my general recommendation. sequentially rather than in parallel?
Okay, so I will share my experience. So when I first went back to Taiwan, then I decided to, I was allocated to the Department of Research in my hospital. So I only have 2 clinic per week and a half day of surgery, and the rest will go to research. So I'm currently having an administrative role on the Department of Research. So I think that's a very good a way for me to have a better arrangement of my research time, and also I'm also having full-time, so-called full-time faculty position in the university, so that I have a chance to get a master and a PhD student regularly from the university, and then so that also strengthens my my lab. So I would say it would be like the package that you discuss with your hospital or the university, and then it really depends on how much time you want to invest in research, or maybe how much salary you want to get. If the salary is not that the key for you, maybe you can discuss, I mean, in the beginning, you can discuss a more research-oriented package to your hospital or university. But of course, that differs from many countries. So for me, that was the case. So I have a good amount of research time so far, and then a balanced surgical or clinical time. Yes, thank you.
Does anyone else have another question?
We should let Hubert go to sleep.
No, I'm okay. Don't worry.
Well, I think we're anyways coming to the end with the time here, but thank you to all of our speakers for sharing their time and their insights tonight. Thank you to all the audience for joining us. I hope everyone's leaving with some new ideas about how to start or strengthen a collaboration. And also on behalf of our ARO Clinician-Scientists Committee, thank you again for being here. And I look forward to continue these discussions, hopefully in the future. Have a good night, everyone.
All right. Thanks, Judith.
All right.
Thank you,
Thanks a
Judith.
lot.
Bye.
Great session. Thank you.
Thank you.
Thank you.